The topic dealt here is mapping genomes by means of linkage maps. Genes are arranged on chromosomes. Gene mapping is a method or technique used for sequential arrangement of genes on the chromosomes. It is composed of many molecular markers. In some cases, the gene itself is used as a marker. Gene mapping helps in locating a specific gene to a particular region in the chromosome and determine the relative distances between the genes and the molecular markers used. There are two different approaches for mapping a genome. A. Genetic mapping. B. Physical mapping. In genetic mapping, distances are measured in recombination frequencies, while in physical mapping it is measured in base pair. The figure here shows the different approaches made to construct a genetic map and a physical map. A genetic map can be made by using linkage maps and DNA markers of different types. Physical maps are made using restriction maps, sequence tag mapping, fish techniques, radiation hybrid maps, contage maps. The genetic map is created using a large population of segregating population, while a physical map relies on a large DNA fragment ordered along the chromosome. When combined, the genetic map and the physical map help in disappearing the locations of genes along the chromosomes. Genetic mapping. Genetic mapping involves techniques to construct maps with positions of genes on chromosomes. It includes crossbreeding experiments or pedigree analysis. It was T.H. Morgan and his students who came up with the idea that recombination frequency rates could be correlated with distance between the genes and can be used to determine gene order on chromosomes. The first genetic maps created used phenotypic markers like eye color, wing pattern, and height. The main limitations of these markers are that not all phenotypes are visual. The basic principle of genetic mapping is that genes are located on chromosomes. Genes which are close to each other tend to move together during gamete formation. That is, these genes are linked. If genes segregate independently, it can be interpreted that the two genes are far apart on the chromosome or on different chromosomes. That is, the genes are unlinked. Thus, during gamete formation, two types of gametes are produced. One, parental gametes, and second, recombinant gametes formed due to crossing over. The figure here shows chromosome segregation during meiosis in case of crossing over as well as without crossing over. A test cross is generally done to demonstrate whether the genes on the chromosomes are linked or not. Alleles segregate during meiosis. If alleles are completely linked and are in coupling phase, only parental types would be formed in equal proportions, that is 50% each. If the alleles assort independently, equal proportions of parental and recombinants would be observed. That is 25% of each type. In case of partial linkage, varying proportions of parental and recombinant progenies are observed. More than 50% of the progenies would be parental types. Partial linkage groups are used for making linkage maps. Both single crossover and double crossovers can be studied. Data of parental and recombinant progenies of a large segregating population is recorded and the recombination frequencies are calculated. The earlier figure showed single crossover with two linked loci. Here, three linked loci are present and it shows both single crossover and double crossover. The thing to be noted here is the recombinant chromosomes resulting from the double crossover have only the middle gene altered. Calculating recombinant frequency. Recombinant frequency is the ratio of number of recombinant progenies to the total number of progenies. If the recombinant frequency is equal to or more than 50%, the genes are considered to be not linked. Closer the two genes, lesser would be the recombinant frequency percentage. Distance between the genes is measured in map units. One map unit is equal to 1% recombinant frequency. Genetic distances measured with recombinant frequency rates are approximately additive. Example, let's consider three genes A, B, and C. After calculation of recombinant frequency, the recombinant frequency between A and B and C are estimated to be 5 and 10 map unit respectively. Then the distance between A and C is summation of distance between A, B and B, C, that is 15 map unit. The genetic map provides the order of genes on a chromosome and the approximate distance from one gene to another based on recombinant frequencies. The occurrence of crossover is not an independent event. The occurrence of the first crossover inhibits additional crossovers in the same region. This is known as interference. Interference is calculated by the formula 1 minus coefficient of coincidence, where coefficient of coincidence is the ratio of number of observed double crossovers to the number of expected double crossovers. Now, if the distance between A and C is to be calculated, it is the sum total of distance between AB and BC, that is x plus y. However, due to the phenomena of interference, the distance between the genes in case of multiple crossover is always underestimated. This is corrected by means of mapping function calculated using Poisson's distribution. As of now, we won't go into much details of these. Steps to determine gene order in a three-point cross. I will explain it with an example on the extreme right. Here, a test cross is made between wild-type Drosophila and mutant with scarlet eye color, ebony body color, and spineless. The gene order of the gene is not known. The progeny data is recorded and analyzed. First, identify the two phenotypes which are maximum in number. In this case, it is wild type and scarlet ebony spineless with 283 and 278 progenies respectively. These are the non-recombinants or the parental types. Next, identify the two phenotypes with the least number of progenies. Here it is spineless and the other scarlet ebony with 5 and 3 individuals respectively. These are the double crossover recombinant types. Next, compare the non-recombinant progenies and the double crossover progenies. All except one character would be different in the two groups. In this case, the spineless gene. Thus, this gene is encoded in the middle. Please refer slide 7 to understand the progenies obtained from single crossover and double crossover. Therefore, the gene order in this case is scarlet, spineless, and ebony, or ebony, ebony, spineless, scarlet. Once the gene order is known, the recombinant frequency is calculated for each gene pair. Recombinant co-frequency is sum of single crossover between the two genes in consideration plus double crossover progenies divided by total number of progenies. In this way, the map distances between the genes as well as the gene order is known. The figure here shows the linkage map of Drosophila. The first linkage map of Drosophila was constructed in 1916 by using the recombination frequency data collected by Bridges. Thank you.